Dear students, in the last lecture we had studied the structural and functional organization of bacteria in detail. Let us have a quick revision of what we had studied in the last lecture. I have told you about the sizes of the bacteria. The smallest one, they have about zero point 0.1 micrometer diameter, and largest they are 16 to 6 micron, and Rickettsia and Chlamydia and Spirulum volutens were respectively their names. In the sizes we had studied that they are of spherical shape, which are called as cocci, rod shape bacillus, and when they are of spiral shape they are called as they have a spiral kind of geometry, and mycelial forms also exist. We also studied some of the examples of spherical, rod, spiral and mycelial shaped bacteria. We had studied the structure of bacteria, and in which we had studied the cytosol, and the cytosol contains enzymes, but no membrane bound organelle was cited in the cytoplasm, which is the mark of prokaryotes. The ribosomes were 70 Swedberg unit type, and no Golgi, no endoplasmic reticulum, which was present in the eukaryotes, was cited in the bacteria. The protein synthetic mechanism in bacteria is hampered by two antibiotics, namely streptomycin and chloramphenicol. So whenever any question comes to you, what is the mode of action of these two antibiotics, the answer should be that they inhibits the protein synthetic mechanism of bacteria. In the cytoplasm, we have membrane-bound inclusion bodies, which contains glycogens, and others are carboxysomes, chlorosomes, and magnetosomes. These have membrane which is slightly different from the unit membrane. Membraneless granules all are volutin granules, polyglucan granules, polybutyrhydroxybutyrate granules. This poly-beta hydroxybutyrate granule is the reserve food material when no nitrogen is present in the environment. Photosynthesis is done by thylakoids, and we have chromatophores, which contains the membrane bound vesicles, which contains the photosynthetic enzymes such as cytochromes, carotenoids, also contain their chlorophyll, which is called as bacteriochlorophylls, the enzymes of etc, etc. Then another important structure which we talked about was nucleoid. It is the chromosome of bacteria, and it is about 100 micrometer in diameter and usually remain attached to the plasma membrane. Plasmid, another unique feature, which is a small circular DNA, which does not carry much importance apart from having some antibiotic resistant genes. If we take out the plasmid from the bacterial cell, it won't affect its survival. And it is the plasmids, I told you in previous lecture, they are meant for they affords the sexual dimorphism or maleness to the bacterial cell in which they are present. The mesosome, which is the in-pushing of plasma membrane, is actually plays the role of mitochondria in the bacteria. Apart from DNA replication, the plasma membrane also play a role in septum formation during the fission, and electron transport chain mechanism is located in the mesosome, which is the in-pushing of plasma membrane. Now, in the previous lecture, we forgot to study about the flagella and pili. Today, we'll take up flagella and pili. And that flagella are a longer structure having dimension of 5 to 15 micrometers, and they are the motility organelles. They are entirely made up of protein flagellin, and their number varies uh, in bacteria. When only one flagella is present at one end, such a flagellation is called as monotrichous. When uh, 3 4 flagella they are present at one end, such a bacterial cell is such a flagellation is called as lophotrichous. When both ends of the bacteria they have flagella, it is called as amphitrichous. And when all the sides of the bacteria they have having flagella, it is the condition or flagellation is termed as peritrichous. So flagella have only one function to perform, which is motility in the liquid medium. Now come to pili and fimri. They are also small fibrous structure, but they don't have any locomotory capacity. They, in fact, they do not provide any locomotory ability to the bacterial cell. They are made up of a protein called as pilin. They are smaller than flagella, thinner than flagella, and have no role in the motility of the bacterial cell. But they have one function to perform, that is they form bridge between two conjugating cells and confers the sexual dimorphism. The sexual dimorphism is conferred to the bacteria, as I have told you in the previous slide, that whenever a bacterial cell is having the plasmid, it is called as male and it must have pilus. This pilus is a transient structure or temporary structure which gets dissolved after some time, after the passage of the genes of plasmids. Then cell wall is made up of peptidoglycan, and when bacteria they are treated with lysosome, the cell wall get lysed and the wall-less bacterial forms they are termed as L forms. These have the capacity to regenerate the cell wall again. Then the outermost structure is capsule, which is made up of polysaccharide. It prevents the bacteria from attacks of the phagocytes and prevents the rapid loss of water, rapid loss and gain of water in the bacterial cell. The capsule also facilitates the ion exchange, and the virulence of the bacteria is the virulent capacity of the bacteria is because of the presence of capsule. Now today we are going to take up gram positive and gram negative bacteria also, and before that let us come to spores. Some bacteria, not all, form spores in unfavorable condition like Bacillus and Clostridium. They afford the resisting capacity to the spores, and this resistance capacity is afforded to the bacterial cell by calcium dipicolinate. The outermost layer of the spore is exosporium, and it is layered one, also called as spore coat or cortex. The protoplasm inside has calcium dipicolinate. Then comes the sexual dimorphism. The F episome, which is also called as F factor or also called as plasmid, affords the sexual dimorphism to the bacterial cell. The bacterial cell which has F episome must have F pili, and by citing F pili we can deduce that the bacteria has maleness. F minus depicts the recipient nature of the bacteria, that means the femaleness. Whenever two bacteria conjugates, F plus and F minus, they come in the process of conjugation. Bacteria occasionally enters into HFR state. Now what is this HFR state? When the bacterial plasmid, it integrates with the normal nucleoid, the strain enters into a state which is called as high frequency state or HFR state. So this was regarding the structure. Now come to the difference between gram positive and gram negative bacteria. Uh, First character we take up in studying the difference between gram positive and gram negative bacteria is homogeneity. The gram positive bacterial cell wall is more homogeneous and less complex and is single layered. You just see this diagram over here at the top in left one, that the gram positive bacterial cell wall is very homogeneous, whereas you can see the heterogeneity of the gram negative bacterial cell wall. In the cell wall of gram positive bacteria, you see a cell wall laid upon the cell membrane, and it is made up of peptidoglycan, and the peptidoglycan is having its contribution to up to 80%. Then we have teichoic acid also in the gram positive bacteria. So as far as the homogeneity is concerned, gram positive bacteria are more homogeneous, less complex and single layered, whereas converse in gram negative bacteria, they are less homogeneous, more complex and multi-layered. Again, you can see as pictured in gram negative bacterial cell wall, it is made up of lipopolysaccharide, phospholipids, lipoproteins, peptidoglycans. That means much of heterogeneity is there, and it is multi-layered. The thickness is 20 to 30 nanometer, whereas gram positive negative bacteria, they have a thickness of 10 to 15 nanometer. Then not much difference is there in the thickness. And the chemical nature of the gram positive bacteria is that they have peptidoglycans, and the proportion is 20 to 80%, and other components are proteins, polysaccharides and teichoic acid. The gram positive bacteria, they are so sensitive to lysosome that their cell wall gets sliced by the lysozyme which is present in the tear, and they are also more susceptible to the antibiotic penicillin. Whereas the gram negative bacteria, they have peptidoglycan in the proportion of 5 to 15%, lipopolysaccharide, major constituent of gram negative bacterial cell wall, it is of 50%, then phospholipids 35% and proteins 50%, very heterogeneous. The surface antigen in gram positive bacteria is teichoic acid, whereas the surface antigen of gram negative bacteria is lipopolysaccharides. The gram positive bacteria, as far as toxins are concerned, they form exotoxins, and the gram negative bacteria, they forms endotoxins. The examples of gram positive bacteria which you have to memorize is are Bacillus anthracis, Bacillus coccus, Clostridium tetani, Clostridium botulinum, Diplococcus pneumoniae, diphtheria, Streptococcus, Staphylococcus. You can have aid to memory to learn these names as BB, CC, DD and SS. BB means Bacillus and anthracis and Bacilliococcus, and we have Clostridium tetani and Clostridium botulinum, Diplococcus pneumoniae, diphtheria, Streptococcus and Staphylococcus. The examples of gram-negative bacteria are Shigella, Salmonella, Spirulum and Pseudomonas. You can also see the 8 to memory for this: S to the power 4 n to the power 2 BVE. Shigella, Salmonella, Spirulum, Pseudomonas, Neisseria meningitidis, and Neisseria gonorrhoeae, Bordetella, Vibrio cholerae, and Escherichia coli. So students will take up these gram negative and gram positive bacteria and their staining purpose in the upcoming lectures. And this is all for this lecture. Thank you very much.